The overall aim of this experiment is to assess craving for sucrose in rats after either a short or protracted period of forced abstinence from sucrose self-administration. This is accomplished by first allowing rats to self-administer sucrose in 10 daily self-administration sessions. Then, the rats are placed into a period of forced abstinence from sucrose self-administration, simply by keeping them in their home cages for either 1 or 30 days. The final step of the procedure is to return rats to the self-administration environment and allow them to respond to cues that were previously explicitly associated with sucrose self-administration, but not for sucrose itself. Ultimately, the results show how the vigor of responding for sucrose-paired cues varies according to the length of forced abstinence. As we have previously reported, sucrose craving increases or incubates over a month of forced abstinence from sucrose self-administration. Experiments may be conducted using this procedure to investigate behavioral and neurobiological substrates of the incubation of craving.
The main advantage of this technique over existing methods is that the length of time following the final self-administration session can vary.
This method can help answer key questions in the field of addiction research, in particular how the length of a period of forced abstinence affects craving.
The implications of using this technique extend toward therapy for relapse behaviors, in particular as a vast majority of individuals have relapsed after an extended period of abstinence.
Though this method can provide insight into sugar craving, it can also be applied to other rewards such as drugs of abuse.
This study